Dr. Ferrante completed his PhD in computer sciences at Université Paris-Saclay and the INRIA in Paris, France, and worked as a postdoctoral researcher at Imperial College London in the UK. He has also been a visiting student at the CVGL lab at Stanford University, a Fulbright visiting researcher at the A. Martino Center for Biomedical Imaging at Harvard Medical School in Boston, a DATAIA invited professor position at MICIS lab at Centrale Supélé, Université Paris-Saclay. He returned to Argentina in 2017, holding a faculty researcher position at Argentina's National Research Council, CONICET. He regularly serves as a member of the program committee of important medical imaging conferences like MICAI, MIDL, ISBI, and IPMI, and he has organized several workshops in this context. He leads the Machine Learning for Biomedical Imaging Computing workshop research line in the Research Institute for Signals, System, and Computational Intelligence, SINC. Dr. Ferrante received the Young Researcher Award from the National Academy of Sciences in Argentina, the Mercosur Science and Technology Award, and the Google Award for Inclusion Research for his scientific projects related to AI for medical image computing. His research interests span artificial intelligence and biomedical image analysis, currently focusing on fairness, domain adaptation and generalization, calibration, and anatomical segmentation of medical images. Thank you for coming, Dr. Ferrante. I'm super excited to have you here, and this is like a very interesting topic to me, so I'm excited to hear your talk.
Well, thank you very much, Juan Pablo, for the invitation. I'm glad to be here talking today about some of the work we have been doing during the last years, basically on two different areas. On the one side, we will be talking a lot about segmentation, which is a really important problem for medical imaging in general. And also, we will try to link this with the fairness aspect of this. Okay, so the talk will be kind of divided maybe in two parts. I know that many of the people in the seminar maybe are coming from the brain side. The talk is not going to be focusing on brain. It's mostly going to be focusing on X-ray images, okay, of the chest. But basically, I think that most of the techniques I'll be describing here apply directly to the context of brain images. Actually, I also work on brain images, but for this talk, I decided to focus on this project in particular. So first, I'll start saying thank you to all the people that has collaborated to the different research that I'll be presenting today. Many of them are here involved. This is my group in Argentina, where I am currently located. Okay, great. So today I'll be talking about semantic segmentation. Okay, so basically, medical image segmentation. So basically segmenting organs in medical images. And this is a really fundamental problem for our community, where we basically want to contour some kind of organ. In this case, this can be lungs, but it could also be in brain MR, for example. We could be contouring different tissues of the brain, like white matter, gray matter, or we could also be contouring subcortical structures. So basically, we want to segment organs somehow. And I like starting this presentation making this difference between what it means to segment organs in medical images and what it means to segment just images in the wild. In case of standard natural images, like here we have a cat, we have a cow, what usually happens is that we have a lot of data. Okay, so if you look at the computer vision data sets, these are huge. But the problem with these data sets or this particular task is that it has low regularity, in the sense that if you look at a cat, the cat will be completely rotated in different parts of the image, whatever. Now, when we look at medical images, in general, we have much less data. Like usually our data sets are order of magnitude smaller than the huge computer vision data sets. So that makes it probably more difficult. However, there is an aspect that we will be trying to take advantage of today during this talk, that is the high regularity that these images have. High regularity in the sense that if you look at the lungs, they kind of always look the same. They have a certain structure. And when we talk about anatomical segmentation, segmenting organs, what we will say is that these structures live in some kind of anatomical space, and we will be talking about the concept of anatomical plausibility. And basically, that means that the segmentation I'm doing somehow looks like a line. For example, if we think about, I don't know, here, segmenting lungs or segmenting heart in an MR image, well, these are clearly examples of non-anatomically plausible segmentations in the sense that they have, for example, a hole in the middle of the heart. That cannot happen. There are topological constraints that are there when we deal with medical images in general. And so much of this talk will be about how can we take advantage of this regularity and impose constraints in the space of solutions so that we guarantee that the segmentations we get are anatomically plausible. Okay, that's more or less the first part of this talk. Here is, for example, when segmenting with free surfer, for example, the white matter surface, you can see that there are topological errors there that shouldn't be there. So that means this is not anatomically plausible. How can we encourage in our deep learning methods such anatomical plausibility? First of all, why does it happen? In general, the deep learning models that are state-of-the-art, or the deep learning models, sorry, that were state-of-the-art until some time ago, like CNNs or even transformers, have local support in the sense that they, even if transformers have like a bigger, like a wider kind of receptive field, if you want, but you have usually local support in the sense that one of the predictions is going to be influenced by a certain area of the prediction of the image. Okay, if we think about convolutions have by construction local support. Basically, if I'm predicting, I don't know, something here, what's the label for the heart? Well, this label for the heart will be influenced by some area determined by the receptive field of the network. And that makes that sometimes, I don't know, if the network is looking at the white area and all surrounded by white pixels, well, it may not know whether this is background or being really in the middle of the heart, okay? This fact is kind of stressed by the fact that the loss term that we usually use when we train segmentation models, like the unit, for example, is defined at the pixel level. And what it means to be defined at the pixel level is that the loss term doesn't care where the errors are happening. If they are happening here or there, everything is going to be the same. And so there is no way in which we can incorporate these kind of global shape constraints, which may help us to guarantee somehow the global consistency of this segmentation. By the way, if there are questions, I don't know if this is, maybe you can just jump in and ask. I don't have any problem with that, or we can leave them for the end. But if you want to open the mic and just ask, I'm happy with that as well, okay? So this is basically the problem. And then one of the questions I started to work on when I was at Imperial College in the postdoc was, how can we take advantage of this high data regularity to encourage anatomical plausibility in our segmentations? And this was a paper by Ozan Oktay, one of the PhD students that was there at the time, and I had the opportunity to collaborate with him and learn a lot from him, where he proposed in his thesis this anatomically constrained neural network concept. And it's a nice idea, and even though today it may not...
Fine.
Sorry?
I'm listening to a seminar anyways. Hold on.
There is someone talking. I think it's Ethan. No. It's okay? Okay. So I continue? And so the idea with anatomically constrained neural networks is we want to learn an embedding which will constrain the global information about the shape and topology. How can we basically take our Oh, I think I was mute. That is okay? Can you hear me?
Yeah, we can hear you now.
Yep, we can hear you.
Okay, perfect. So the idea is we want to learn some kind of low dimensional representation from the segmentation masks that contains global information about the segmentation, and this will help us to impose some kind of global constraint in the segmentation problem. And at the time, one of the ways we knew to learn low-dimensional representations was to use autoencoders, okay? And most of you may be familiar with autoencoders, but basically the idea is that we will train actually a denoising autoencoder, basically a neural architecture that has a bottleneck at some part. And so the input to the network is basically the segmentation, and we force it to go through this bottleneck. Then from this bottleneck, we reconstruct the original segmentation, and we train this model using a loss function that basically imposes or teach the model to reconstruct it from the low-dimensional representation. This forces the model to learn useful information in this low-dimensional latent embedding that will have information about the whole structure, because actually this embedding here is fully connected, so it has complete receptive field. It will know about the whole image. We actually use a denoising autoencoder that is basically just a modified version where you introduce noise at the input and you train the model to denoise, okay? So basically in a denoising autoencoder, you will have your image with the noise, and you will ask the model to basically recover the original image there. Adding noise helps to learn kind of more softer embeddings, okay, with better support somehow. And the idea is that this H is going to be our embedding, okay? So we have what we call the encoder here, the decoder, and the embedding is going to be the output of our encoder. And this will be trained using a loss function, like, for example, here, the mean squared error, okay? So we take our noisy image, we pass it through the encoder here, then through the decoder, and finally we take the difference with the original image, with the original segmentation. Importantly, we do that using only segmentation masks, okay? The moment in which we learn the embeddings is going to be only done using the segmentation masks. No image information is going to be there. So you can think about brain masks for different structures in the brain. It could be the same. Now, it's interesting to analyze what happened with this latent space that will help us then to impose these kind of global constraints. Because if we look at the segmentations that we produce, let's say I have a model that is producing this segmentation here, and this would be the ground truth segmentation, for example. Then we can compute two types of loss functions. For example, if I compute a loss function at the pixel level, that can be simply the cross entropy at the pixel level, for example, okay? And this will have local information somehow, because it's going to be looking pixel at pixel, not at the global picture. In this anatomically constrained neural network paper, what Ozan proposed was to use a global loss defined as the distance in the embedding space. So we are not now looking at pixels differences, but we take the segmentation that we produce, we pass it through the encoder, we take the ground truth that we are comparing with, we also pass it through the encoder, and we take the difference between these two embeddings. And this is what is here. It's basically the distance between the latent embeddings for the prediction of my network and the ground truth. And we will use a combination of these two losses to train the model. So before, let's say we are segmenting, for example, here, a heart. So this heart is just going to go through the segmentation network and produce a segmentation map, and we will train it using cross entropy, for example. Now, when we go with the anatomically constrained neural networks, what we do is basically pre-train an autoencoder using only segmentation masks. This is the autoencoder I'll be training here. And then, when I train my segmentation network itself, my loss function is going to be composed of two terms. The first term is going to be the cross entropy part at the pixel level, the typical one that you use. So you could use the dice loss, for example, which is another loss that we usually use. But I will combine it with the global loss that I was telling you. So basically, the output segmentation is going to go through the encoder. This encoder that was pre-trained will produce a latent embedding. I will do the same with the ground truth and take the difference between those two codes. The nice thing about that is that, for example, if you look at results for different type of images, look at here, for example, this is a pretty noisy and corrupted heart image. It's a heart MR, where there is noise between the different slices, okay? There is motion, sorry. If you look at the predictions from a model that basically segments slice by slice, you will get something like this. Now, when you look at the prediction of a model that incorporated this anatomical constraint that looks at the global level, it will look like that. Much softer and much more similar to the ground truth that we have here in green. The same, for example, for an ultrasound image that is really noisy, and by using the ACNN, you will get this kind of improvements. Actually, also numerically, you will see that, particularly in the Hausdorff distance, that will be reduced a lot by using this idea of anatomical constraints. So we saw that this idea was really, really interesting. And when I arrived to Argentina, I wondered, okay, can we extend the same idea, but to a different context, for example, to the context of image registration? And in image registration, what you have is basically two images that you want to align, okay? And when you want to align the images, it can happen that you deform one of the images too much, in case you are dealing with deformable registration, okay? Deformable registration allows the image to be deformed to match the other image. And this is used in many cases, particularly in brain image, we use it a lot to align, for example, different brains before doing some kind of population analysis, or also for multi-atlas segmentation. There are many tasks in which you will use this kind of registration. But you have these problems that sometimes you deform the source image too much and you make it non-anatomically plausible. So we wonder whether we could take advantage of the same ideas that we exploited before and use them in the context of medical image registration. Sorry, that we can do, and there are many ways, but this is kind of the standard way of doing medical image registration using deep learning. So here we have a network that is trained to register two images. And in general, the network works as follows. You take the source image, the image you will deform, you take the target image, and you concatenate them, and you give them to a network, like this one here, that will produce a deformation field at the end. This deformation field is basically indicating where should every pixel be moved from the source image to match the target. So you take this deformation field and you warp this source image, and you produce the warped source image that should be similar to the target. And you compute a loss function based on some similarity metric that can be, for example, the sum of absolute differences or something like that. And then you also have usually a regularization. And this model is trained to basically reduce this similarity metric. Now, what we proposed was to basically add an extra part in the network where you not only take the images, but you also take segmentation masks and you apply exactly the same idea we were discussing before for segmentation, but now to regularize the organs so that they are not folded, they are not basically destructed during the registration process. And this worked pretty well. You also have a pixel level loss and a global loss, and seems to work pretty well. And in this paper, we did this interesting analysis where we wonder, okay, why these ideas are actually working. And we did this. Look, I think this can shed some light on why these models are working. Here I have a reference segmentation. So basically the segmentation I would like to get. And what we did, it was to generate different versions of this segmentation by removing pixels. Okay, so we swapped foreground pixels to background, sorry. And we swapped a fixed number of pixels. So we took 120 pixels and we swapped them to background. Okay, so if you look at these four images, they are all modified versions of this first one. But if you look here, we did some anatomically plausible modifications here in the left. We basically eroded the pixels from the border. But if you look at this, you will say, yeah, this looks like a good segmentation or an anatomically plausible segmentation. Now, when you look at the other ones, you can see that we removed, for example, a part of the lung, a part of the other lung, a part of the heart. These are clearly non-anatomically plausible versions. And now what we did was to compute the pixel level loss, the standard loss, the loss that uses the cross entropy at the pixel level. And of course, this was constant. Why? Well, because we modified in all of this the same number of pixels. Now, when you look at the other loss, the one based on the embeddings that we were proposing, we observe that actually this one has a much lower loss compared, for example, to this one here or these other modified versions, where we are removing parts of the organs. So these two losses somehow are complementary. In cases where the pixel level loss would be constant, our loss would tell you this segmentation is better than this one. And that's how the model, by combining the two when you are training it, is basically achieving more anatomical plausibility. And if you are interested in this, you can go and check the paper. There are many experiments. Of course, this was not done on a single image. This was done on hundreds of images, and here we are showing the statistics for that. Okay, but then we continue with this line of research, and we say, yeah, this is a really cool idea. It helps us to get anatomically plausible segmentations. But the problem is that we need to use it during training. So when we are training our network, we need to modify the loss function and do all this machinery, adding an extra loss, etc. So we say, is there any way in which we can take arbitrary segmentation? So let's say I have a method based on whatever technique you want that gives you a segmentation that is not really good. It's not really anatomically plausible. Like in this case here, these lungs are completely corrupt. And can we do something to recover the anatomically plausible version of this segmentation that is basically closer to this segmentation? And that was how we proposed this paper that is called Post-Denoising Autoencoders, where we took advantage of these autoencoders that we were talking about, but we used them as post-processing step. You know, for people coming from the old computer vision school, you know that before deep learning and everything, we were doing a lot of post-processing. Sometimes we were getting like an initial result, then we were refining it using some kind of refinement, for example, conditional random fields or stuff like that, were helping us to improve the segmentation a little bit. So this goes a little bit on that direction. And the idea is pretty simple. What we will do is to train a denoising autoencoder using only anatomically plausible masks. Okay, so this denoising autoencoder will be trained using only masks that, when reconstructed, they are anatomically plausible. That means that the autoencoder, the only thing it knows how to reconstruct is masks that are anatomically plausible. So the hypothesis was that when I input here something that is non-anatomically plausible, this autoencoder will basically help us to recover the anatomically plausible version of the segmentation that is closest to it. And the idea, I mean, when we talk about denoising autoencoders, you know that there is this hypothesis that is called the manifold assumption that in general is there when we talk about representation learning methods like in this case, the autoencoders, that tells us that given a set of, for example, lung segmentations, images of lungs, they all live in a low dimensional manifold. If we can learn this low dimensional manifold, then we can map any segmentation to this low dimensional manifold, and when we reconstruct from there, we will reconstruct the anatomical version because it's basically what is living in this manifold. And this is somehow what a denoising autoencoder is doing, right? So here in this line, let's say this is the manifold where all anatomically plausible lungs are living. When I take a corrupted anatomical segmentation like a lung here, this is somehow outside of this manifold. But when I pass it through the autoencoder, I force it to be projected to the manifold and then reconstruct. And this is the effect we are looking for. And with this simple idea, just training a denoising autoencoder using anatomically plausible masks, we managed to obtain these results. Look, here we have images that we were trying to segment. Here we have predictions from different segmentation models, okay? This is, for example, coming from a random forest model. This is from a unit that was trained for just a few epochs. This is a unit that was trained for more epochs. And here we have the result that we would obtain, for example, if we use some of the standard at the time post-processing methods like a CRF. You can see that there is an improvement somehow because it homogenizes the labels, but it's not really making them anatomically plausible. Now, when you take these segmentations and you pass them through our post-DAE model, you get this thing here, the fourth row. And the ground truth is the fifth. So what you can see actually is that you recover a pretty anatomically plausible segmentation that actually looks much like the ground truth. And the interesting thing is that here I'm not involving any type of image information. This is all based on labels, okay? The autoencoder is not really looking at the image, it's just looking at the labels. And of course, you get also some improvements in terms of dice coefficient, also in terms of Hausdorff distance. There are more details there in the paper, but I like showing this video because it kind of shows that we are not doing cherry picking. So this is the image. This is the initial segmentation horrible, like the ones you can see here. This is what you recover with our post-DAE, and this is the ground truth here on the left. So you can see that even though it's not perfect, of course, the result looks much like a land that they were looking before. And this is Agostina, the first author of the paper, and the code is actually available. You can look there. The trick with this was how to train this autoencoder, how to generate this noise. It has to be noise that is somehow realistic. So you can go and check out the details there. And then we also get these ideas about, okay, denoising is pretty useful to recover anatomical plausibility. Well, when I visited MGH a couple of years ago, this was a collaboration done with people at the MGH where they say, hey, if denoising is useful as a post-processing step, then it can also be useful during training. Because maybe if I have, for example, images for which I don't have any kind of ground truth, okay? Imagine I have a lot of images without annotations, but I want to use them somehow in my training process. So what you can do is train your model, use the images for which you have annotations. And for those where you don't have annotations, make predictions using the model that you are training, which of course is not going to be correct, the prediction, but it's going to be somehow good, and pass it through a denoiser, improve the quality using this idea of post-DAE during training, and backpropagate the loss for these images. That allows you to use images that are not labeled, so unlabeled images, but this helps to the whole learning because at the end of the day, you end up using more images than before. And this was, these worked pretty well. This is work done by Xian Yong and was published last year in TPAMI. And you can go and check the details on the paper. Okay, so far I've been talking about segmentations based on pixels. But it's a whole different world out there where segmentations may also be based on landmarks. And this is paper we published at MICA in 2021, then an extended version at TMI. So instead of looking at this type of segmentations, pixel level, we want to now look at graphs that represent the organs. And these graphs are defined by landmarks, okay? And these landmarks are somehow, some of them have anatomical correspondences. How can we train a network to, given one of these images, produce this landmark? And this is in 2D, you will get these contours. But if you think, for example, in 3D, in brain images, what you will get is a surface, okay? It may be a surface, for example, in the contours of the white matter or gray matter, things that are actually produced by free surfer, okay? So how can we design an architecture that, given an image, gives us a graph? And this is not a straightforward problem because here we are changing the structure of the data. When we go from images to pixels, this is somehow easier because there is still a grid structure. But here now we go to something completely different. we have a restriction in our model that is all the data samples that we use for training this network have to have the same number of nodes in the graph with correspondences. That means that, for example, for all the subjects in my database, the node number 0 has to be there in the apex of the left line, for example. And the 21 has to be here on the bottom part of the left line. Okay, there is this correspondence. This is a restriction, but it's usually something that happens in medical data sets because we may be building these data sets using atlases. And when you do that using atlases, this is something that you get for free. So, okay, there is this case. How can we now construct a model for doing that? And we first constructed graphs. So here the graph is going to be a graph G, where the vertex are going to be the set of nodes, which is going to be fixed. All the subjects are going to have the same set of nodes. Adjacency matrix is going to have 0 or 1, depending on whether the nodes are connected or not. And basically, the difference between the different subjects will be given by X, that is a matrix that indicates the position of every vertex. Okay? It's going to be like a function where every vertex assigns a 2D position in this case. If we are talking about the surface in 3D, then this will be a 3D position. And so what we proposed, because we liked a lot autoencoders, was to also base our model on that, but we created this hybrid architecture in the following sense. We first pre-train an autoencoder using images, as I was showing you before. But then we also pre-train an autoencoder on graphs. You know that we can learn from graph structures. This is the geometric deep learning field, basically teach us how we can do learning on these kind of structures like graph, networks, surfaces, etc. So we can do convolutions on graphs. We can also have neurons that operate on graphs. And so we can train an autoencoder that learns an embedding in the space of graphs. We did that. We trained these two different autoencoders, and then we cut them. And we connected them here through the latent space. And we continued training this model, backpropagating a loss based on the mean square error between the predicted positions and the original and the prediction and the ground truth positions. The interesting thing about this is that it imposes a strong prior on the type of segmentations we will get at the end of the day. Why? Because by construction, this model only knows how to generate things that make sense, that are anatomically plausible, that has things that have a certain number of nodes, where every node is associated to a different anatomical position. So as we will see now, this results in a much more robust model that allows for segmenting structures in different data sets, and we will see that it performed pretty well. So this is the idea. We compare that with different type of decoders. These are not standard or denoising autoencoders. These are called variational autoencoders, okay? These help us learning here kind of probabilistic distribution on the latent space. I'm not going to get in the details of that, but just for people maybe familiar with that. And also here, we use what is called spectral graph convolutions. That is a particular type of convolutions based on the graph Laplacian. Also, I'm not going to get into the details, but there are references there, or you can check our paper. And this is the architecture you end up having. You have your input image. You basically go with the standard convolution pooling, until you get to the latent space, from which then you reshape and start convolving on the space of graphs. Okay. Well, we did some improvement for that. Again, I think I'm not going to have time, so I want to speed up a little bit to get to the fairness part. But basically here, what we proposed was, in the unit architecture, there is something really important, that are the skip connections. The skip connections help you, that go from the encoder to the decoder, basically. These are like shortcuts that jump like this, and help you to recover detail from the high resolution images as you are reducing the resolution of the feature maps. The same idea, we try to port it to the context of image to graph skip connections. And we created this new model that allows us to implement these skip connections from an image encoder to a graph decoder. Okay? This is not straightforward how to do, and well, I'm not going to get into the details, but If you are interested in that, you can check our EMI paper that appeared in 2022. And well, basically what we showed is with this architecture, we get really good results, competitive even with the NN unit, that is one of the most advanced architectures for this type of tasks. And interestingly, our model always guarantees to have anatomically plausible structures. While if you look at pixel level models, not graph level models, well, you can have this type of hallucinations around here, errors basically, where you are segmenting things as lang, which are not langs. And I think an interesting experiment with this is the robustness experiment we did. So we generated in the images random black squares where basically simulating some kind of anonymization. And we observe how the different pixel level methods work. And if you look here, all of them, the unit, the NN unit, have a lot of trouble with that because basically you are occluding a part of the image, so you don't know exactly what's going to happen in this part. So it tends to assign background. Now, our model, since it has such a strong prior on the structure that has to be segmented, will always provide you with some anatomically plausible structure. And you can think about this in the context of brain structures where these kind of non-topologically correct surfaces were happening with tree surfer. Well, this may be a way in which you can correct for this type of topological errors. Actually, we did this ablation study where, well, this kind of experiment where we increased the size of these occlusion blocks, the black blocks, and we observed that the dice coefficients for our models were pretty constant. Now, if you look at a unit, for example, this starts decreasing a lot because of the errors I was showing you. And this also happens with real occlusions, for example, like those based on peacemakers here. The pixel level models tend to fail, while our model tends to recover the full amount. Now, well, we also extended this to the context of 3D. This is also a preprint that is under evaluation now. And here we basically say, okay, let's try to segment meshes from volumes, as I was telling you. And the method worked pretty well. We tested this idea on cardiac meshes, but same could be used on brain meshes, I think. But yeah, this part I will, I think, just because we observed that the method was working pretty well, okay, in general, in many different type of images, X-ray images. And we say, okay, there is no big database with segmentations of langs. I was telling you that at the beginning of the talk. So can we create a huge database of segmented X-ray images with lungs and heart, where we also provide these anatomical correspondences based on landmarks? And when we started to look at the databases with annotations that were available to train our model, we observed that different datasets, for example, here one coming from Hospital A, Hospital B, and different type of hospitals, were coming with different type of annotations. For example, in one case, you only had lungs. In the other case, you had lungs and heart. In the other case, you had lungs, heart, and clavicles. And we say, okay, let's train a model with all these databases and do inference with that model. So let's say I have these 4 different hospitals. Images in these 2 hospitals are annotated with lungs. In these 2 hospitals are annotated with lungs and heart, and here also with clavicles. If I train a model with that, and I segment the images, I would expect something like that. Because even if the heart is not here, well, I mean, it's not annotated as a label, the heart is there, same for the clavicles. Now, if you take NN unit or a naive unit and you just train it in a naive way, this is what is going to happen. A terrible domain memorization problem. The network will learn from which data center the image was coming, and will basically segment only this structure. And we weren't expecting that. But interestingly, what we observed is that our model by construction was simply all the time segmenting the full structure, like here. I mean, there is a lot more on this story that is written in a paper that we published in ISB, I think last year or or the previous year, where we discussed why is this happening and how we can correct for this problem, even in the pixel level networks. But interestingly, our network was doing pretty well on that. I'm not going to get again here into the details. I'm just checking the time. But basically, it happens because of the latent space. If you look at the latent space learned by our network, compare with the latent space learned by a unit, for example, trained naively, what you see is that the unit will clusterize really well by different hospitals. So creating this effect, kind of remembering, yeah, if this is coming from the JSRT dataset, then it only has lungs, for example. While our network will kind of mix all the datasets together, there is no such a memorization effect. And that's due to the inductive bias of the architecture and also the way we are training. And we say, okay, the model is working pretty well. Let's try then to take all the publicly available X-ray datasets that are out there, because there are actually a lot of X-ray datasets available. Now, the problem is that they don't have annotations. And let's try to create annotations for all these images. And that's how we ended up creating the chest mask dataset. That is a large-scale dataset of anatomical segmentation masks for X-ray images coming from multiple centers. So we produced segmentation masks for about 600,000 images that were not segmented. These were based on this idea of contour landmarks with correspondences across subjects. And interestingly, we computed, because there is no ground truth for this segmentation, right? We are segmenting them, but there is no ground truth for that. How can we tell the users what's the quality of the segmentations that we are producing if we don't have ground truth? So we leverage some work done at Imperial College by Ben Glocker's group called reverse classification accuracy that I will maybe briefly comment in a couple of slides, which allow us to estimate a dice coefficient, estimated dice coefficient per segmentation, even if we don't have ground truth. And so we can use this, we validated basically this index using some annotations that we had. We observed that there was a really nice correlation with the real values. And so we released that as a metric so that people can say, yeah, in these segmentations, I can trust. In these segmentations, I cannot trust. And we also validated some of these with expert radiologists from Argentina, who annotated some of the masks, corrected these masks, and then we computed the dice coefficient with that. And interestingly, we also did some kind of fairness audit for these images without any type of ground truth. And what I mean by fairness? Well, here I'm kind of jumping to the second part of the talk that will be really fast because we are almost on time. But a couple of years ago, I read about this interesting article that was published in Nature, and look at the title of the paper. It was a bit like extreme, but it was saying AI can be sexist and racist. It's time to make it fair. So we were developing AI systems to analyze medical images. I saw this paper and I say, hey, so can we be creating AI systems that are unfair, that work better for one population than the other one? We need to audit for fairness in our systems. So these are some examples I always show. Here is a typical example that was one of the foundational papers in the field, right, by Timnit Gebru and Joy Bulonwini, where they basically show that in facial recognition systems, if you look at from an intersectional perspective to the error rate of face recognition systems that were commercial at the time, developed by Microsoft, by IBM, by different companies, what you could observe is that if you take dark female, dark male, light female, and light male subgroups, intersectionally creating them, the error for dark females for the recognition system was much higher, around 20, 34, 34, when compared to the error for light male, okay, which was 0, 0, 8, 0, 3. That was a huge, this paper had a huge impact in our community. People started to look at the systems from that perspective. And well, we basically started to also look at that in the context of medical imaging. And we have several papers on this topic where we show that AI systems for analyzing X-ray images can also be biased, okay? Can have better performance on men or women, depending sometimes on the data was used to train that. Well, we have several articles discussing this. Another paper as well, other people, sorry, as well has shown that models can be biased. But not only classification systems, also segmentation systems can be biased. So if we think about everything I've been telling up to now can be biased. So actually, if you look, for example, at this paper, in this paper, the group from King's College, Esther Pujol, Anton, and Andrew King, and all their team showed that hard segmentation systems can be biased, with respect to ethnicity, showing a much higher performance on white patients than, for example, Asian or Black patients. And this may be due to underrepresentation in the data sets used, etc. But the interesting thing is that it can also happen with the segmentations I was showing you before, with the lung segmentations and with the heart segmentation. We also observe, when you look at, for example, biases with respect to age, we observe that the Dice coefficient for really young or really old patients, really young here on the left, really old here on the right, tends to be lower than for the rest of the patients. And so we basically have created this huge data set with all the segmentations, and we wanted to audit it for biases. But we didn't have ground truth. So usually when you audit a system for biases, what you do is you train your segmentation model, you evaluate this on the different subgroups, segmentation or whatever group, okay, or whatever algorithm, but in this case, it's a segmentation model. You evaluate it on different subgroups, let's say male and female, and then you compute some fairness metrics. For example, the Dice coefficient in both groups, and you take the gap between that, and you look at the gap. If the gap is too big, that means that the algorithm is working better in one group than the other. Now, for doing that, we need to have ground truth annotations. And in our huge data set, we didn't have. So we basically asked the question on whether we can anticipate biases for segmentation networks in new populations without ground truth. And that is a problem that we call unsupervised bias discovery. That is this idea of discovering clusters of bias subjects in which the system is failing, without any type of ground truth. And we published this paper last year in a workshop at MICAI based on the reverse classification accuracy index that I was telling you. I'm not going to get into the details of the reverse classification accuracy index, but just believe me, it's a way that given an image, I here, and given a prediction, not ground truth, a prediction, the index will tell you how good the segmentation is. Okay, and it's based basically on a comparison with some kind of database that you have for which you have ground truth. Now, using this, we basically showed, and I'm going to skip that to leave some time for the questions, that this index, if we take gaps in terms of this index, this correlates really well with the gaps with respect to the real Dice. And this is what we show in the paper, and we basically were able to perform fairness audits in our segmentation masks without having any type of ground truth. And we observed that, for example, when you look at populations divided by sex, male or females, the segmentations tend to have more or less the same distribution of estimated Dice. Now, something that was really interesting for us was here, when we divided our population between posterior-anterior and anterior-posterior X-ray images. So anterior-posterior images, this is the way the image is taken, right? If the X-ray image is put looking at you or from behind. we observed that there was a strong difference in the distribution. So actually, AP images had much lower dice coefficient in general than the PA images. And when we showed this to the doctors, they told us, yeah, that made complete sense because AP images tend to come from hospitalized patients. They are lying in the bed, so the machine has to be put on top of them. And the way this is done is always the same. And hospitalized patients usually come with different type of artifacts because they have different tubes or cables that do not happen in ambulatory patients that are coming just for consultation and to take the image, which usually are PA images because you take them standing. And so that was really interesting. We could kind of discover a reason here for biases. So the model tend to underperform on images, AP images, and discussing with the doctors, we arrived to the conclusion that this may be due to the fact that there are many more artifacts in the images. And of course, the segmentation quality tends to be lower when there are artifacts because there are occlusions, different type of things. So this is kind of to conclude the talk. So we basically discussed the methodological part of it, some segmentation methods, how can we incorporate anatomical consistency in our predictions. And then we also discussed a little bit about fairness, how important this is to guarantee that the systems we are constructing are fair in the whole population and not only on the majority groups. And then how can we actually even audit for fairness if we don't have ground truth? We have to think about, and this is one of the research lines I'm pursuing right now, how we can perform this kind of unsupervised bias discovery, how we can ensure fairness even without having annotations. So these are the funding bodies that help us achieve this work. And thank you very much.
Thank you very much, Dr. Enzo Ferrante. I'm very sure that every one of us who is opportune to listen to this lecture learned not one, not two, but multiple things that we could also apply into our own various researches. So I would like to open the floor for question and answer from the audience.
I can start.
Okay.
So I have a question for the anatomical plausibility framework that you made. More specifically, the second one with the graph-based method. Could you tell me a bit how, maybe if you were to fine-tune that model to a different domain, how would the process be? Like, would you still have the strong prior with the graph if you were to switch to a different domain?
You mean, for example, if we go to brain images and we
Sure.
work with brain, yeah. So yeah, if you just take the framework as it is, and you, for example, get a data set of MR images with, I don't know, white matter surface, which is represented also by nodes, but it's going to be 3D instead of being 2D. The same kind of strong prior effect will be holding there. Which can be good or bad. I mean, you have to really evaluate the application case in which you are going to use it. For example, here, we have observed that our segmentations always follow the anatomy, okay? But sometimes you may be interested in segmenting in the lungs, for example, not the anatomy, but segmenting the air, because there is some kind of infiltration or something that makes that there is, for example, liquid on certain parts of the lung or stuff like that. And you want to quantify how much is air, how much is not air. And in this case, our segmentation model is not going to be useful because the model is always going to tend to segment the whole lung. Okay, there you may go to a pixel level model. So you really have to think about what's the scenario you are pursuing. In case of, for example, MR images, I believe that this is something we want to evaluate, but we haven't evaluated still, I think this method will be really robust to low resolution images, for example, because if you train it even with with higher resolution images, and then you test it on low resolution image. My intuition is that the model is going to work pretty well when compared to, I don't know, pixel level methods because of this strong prior. But this is something we still have to validate. So we did the 3D extension, and we are working right now on that.
Okay. Thank you.
I have a question, Enzo. First of all, thank you for this talk, and I really enjoyed it. Excellent work. I have a question about the fairness component of the presentation. So in terms of just looking at it, I guess, at a higher level, is there an opportunity to identify a framework for identifying bias, where the bias comes from in fairness, especially within the context of medical imaging? Is it coming from the lack of data, right? Clinical research, like underrepresented minorities and women are severely underrepresented, right, in the clinical research. So is that the reason? Is it the people who annotate the data? Is that where the bias comes from? Or is it the models? So is there, could you just discuss that?
No, I think it's a great point that you're doing there, Mehmet. When I started working on this, the main focus for my research was on underrepresentation and analyzing whether underrepresentation in the training data was one of the reasons. And we observed, of course, that it was one of the reasons. Okay, so if you look at our PNAS paper, really talks about that. Now, we then started to move forward and try to complexify the discussion a little bit and start incorporating these other things you're telling about, models, people themselves. We have an article then in Nature Comms that is called Addressing Fairness of AI in Medical Image or something like that, where we started discussing these points and seeing how data interacts with people and interacts with models as well. So there is not only one reason. It's not only underrepresentation. It may be also, for example, noise level that changes from one group to the other one. So somehow you may have more noisy labels for one group than the other one. Or even the features themselves, like the images, may be different in one group than the other one. Actually, if you look at X-ray images, for example, if you look at male and female images, the breasts in the female images create a shadow that is not there in the male. So actually, the feature extractor that you will use in one case may not be exactly the same than the other one. And even actually the task could be more difficult in one group than the other one. So sometimes, even if you have exactly the same number of male and female images, the model will be underperforming in the female group. Why? Because the task is inherently more difficult. And can we say that the model is unfair because of that? I don't know, because the task is more difficult. So we have to devote more resources to improve the model on this particular group. So it's a really complex discussion, I think, but and I think my way of thinking about it has been evolving since 2020 when we published this paper, actually, where we were only looking at underrepresentation. Then by discussing a lot with different people and stuff, we started realizing about all that. There are really nice articles. We have a group of people that is called the FAMI group, Fairness of AI in Medical Imaging. We started a workshop 2 years ago. That's a virtual workshop. Then we also organized it at MICAI last year. We are gonna organize it again this year. And I think it's a really nice forum for discussing all that. So I really invite you to follow us. There is a newsletter, and yeah, we are pretty active with people from Denmark, the UK, Spain, different places.
We'll definitely do that. Thank you.
Thank you so much, Dr. Enzo. I have a question. What exactly will you tweak in your architecture if you are to work with not anatomical body this time around? I'm talking in terms of tumor, and a tumor that has subregions. You know, we have enhancing tumor, we have necrotic core, and these are not normal anatomical items in the body. So what exactly would you tweak to ensure that your model do the segmentation as expected? That's one. Then two, I also observed that on the chest XMAT, the X-ray database you created, you made use of a platform. Can you tell us more about the platform? We are working on similar projects, and we could borrow one or two ideas from what you
Yeah.
did, and you are able to annotate over 600,000 cases.
For sure. So two things. First, you're really, you're right in the sense that everything I've discussed here, or most of it, it's only valid for anatomical segmentation, not for lesion segmentation, like in case of tumors. Or, for example, if you think about brain images, you can think about white matter hyperintensities or stroke lesions or different type of lesions, which are actually not regular, not as regular as the anatomy. So the whole anatomically plausible thing, it's only going to hold for anatomy segmentation, not for lesions. So there you have to go with the even the reverse classification accuracy index that I explained, that I didn't explain, I just went really fast. But this index that tells you how good the segmentation is without having ground truth, this is not working for lesions. It requires this kind of regularity to work. So there is a lot of space for research. How can we take these ideas to the context of lesions? And then regarding the second part, for the annotations, so this was done in collaboration with Hospital Italiano de Buenos Aires, like Italian hospital in Buenos Aires, where one of my PhD students was working. And the platform she actually installed is this one. It's called Label Studio. So this is a server that you can install somewhere, and then clients can connect through a client server architecture online, basically, to your server. And so you will load the images there, and then annotators will have accounts and will be able to label. It's really powerful framework because it allows, for example, for segmentations like landmark-based segmentations like the one you are seeing here, but also pixel-level segmentations. You can also label classification tasks. It's pretty powerful and open source. So you can just install it and use it. I think it's there is some work to configure it, to set it up, but once it's done, then people can use it. And the nice thing is that you don't need to move the images out of the hospital or something. Like so it's nice in terms of privacy because everything stays in the same LAN network. You don't need to go outside.
Yeah, thank you. I'm glad it is open source. I was about to ask that.
Yeah. I think you can just download it and install your own.
Okay. Do we still have any more questions before we take the survey for today's lecture? Attendees? Okay. At this juncture, I would like to call on Maureen. Maureen, can you quickly share the poll? Just take a quick feedback over what the lecture today has been, and we believe strongly that this will be an opportunity for us to improve on subsequent lecture. Thank you so much, Dr. Ferrante. We can't thank you enough for what you've done for us this evening, and we believe that when next we call on you to come and educate us, would accept our invitation.
Thank you very much for the invitation. I'm glad to have been here, and yeah, let's be in touch.